Okay, so thank you for introduction, and today it's my pleasure to present to you my talk about the dicer and its Hellman domain. So, what is dicer? Dicer is an RNase-free endonuclease nuclease, which is responsible for a critical step in so-called RNA silencing. RNA silencing is a pathway that evolved in eukaryotes as a means to get rid of double-stranded RNA, and most probably it evolved as an antiviral system because that can be the major source of threat. So if you look in this pathway, the role of the dicer is to recognize and bind the double-stranded RNA and to chew it into 20 to 25 nucleotide long fragments, which are eventually loaded on some downstream machinery, which ensures finding of the complementary RNA and its degradation. In 2018, Brenda Bowden and colleagues showed quite a nice movie about invertebrate dicer from Drosophila dicer group. And as we can see, the dicer had actually this L-shaped conformation where L1, L2I, L2, and pincer domains form so-called helicases. Then there are RNase 3A and B catalytic subunits, and at the top of the platform there is Cas domain, which is responsible for binding of double-stranded RNA, either tandem or 3' overhang. This is a processed dicer. So as we can see, the clamping of the helicase and the ATP-dependent threading of the RNA takes place. And once the RNA reaches the Cas domain, the substrate is cleaved. So the distance between Cas domain and the catalytic center ensures the correct length of the product. However, throughout evolution, the mammalian dicer lost its ability to process just any RNA, and became specialized in processing of only these short hairpin-shaped RNAs, which are called microRNA precursors, and are genetically encoded, and through this pathway play an important role in endogenous gene regulation. Mouse is the only exception, up to date known, that has something else, and that is the all-site isoform. So all sites of mouse express such an isoform of dicer protein that can process not only microRNA precursors, but also long double-stranded RNAs of various sources, including viruses, so providing so-called RNA interference activity. Our collaborators demonstrated this in an experiment where they took mice embryonic stem cells, replaced the coding or the gene of dicer with sequence coding the all-site isoform, and measured the levels of RNAi products. And as you can see, the levels in wild type are basically negligible in comparison to the response in the mutant cells. So what is the difference between the two dicers? On this scheme you can see that in all-site isoform there is missing the very N-terminal domain, L1 domain, which also includes these two Volcker A and Volcker B motifs, which are ATP binding motifs. So the final question for our project was, how does L1 domain segregate microRNA biogenesis from RNAi? So in order to tackle this question, our collaborators created mice that would express all-site dicer instead of the full-length dicer, and found out that all of these embryos actually died perinatally, so shortly after getting birth. And as you can see on these micro-CT scans, if you compare the all-site dicer scans to the wild type, you can see there were developmental problems with heart tissue, lung tissue, and even this quite significant pelvic cleft in the head. Later, our collaborators also looked at the microRNA levels and found out that they were completely dysregulated in comparison to the wild type. So now there was a question: is it due to the loss of the whole L1 domain, or just because of the loss of its catalytic function? So they mutated the Volcker A motif, so disabled it from binding the ATP, and as you can see on this comparison, there was a handful of dysregulated RNAs. So this means that, first of all, all-site dicer cannot complement for the full-length dicer in vivo, and secondly, that the catalytic function is not that important as the structural one. And that's what encouraged us to study this problem from a structural point of view. So we employed Cryo-EM, and the plan was quite straightforward: to solve structures of full-length dicer, its all-site isoform, and then of both of these isoforms in a complex with microRNA precursor, and we picked primary 15A. Let's start with the full-length dicer. So as you can see, we could reconstruct around nearly 4 axiom resolution Cryo-EM map, and with help of a lot of local refinement, we could actually quite nicely build the model with high confidence, and this has the correct shape, and it also resembles previously published human dicer. So this was really satisfying for us, and we continued with the all-site isoform. Unfortunately, for that one, we couldn't get any reasonable particles, we couldn't get any 2D classes, and after some playing with modeling and with AlphaFold, we realized that most probably it's due to the high flexibility. Nevertheless, we continued with solving structures of the complexes. So for the complex of full-length dicer with primary 15A, we've got 4.1 axiom resolution map, so here. And if you look at the structure, you can see that the RNA is embedded in the bus domain by its 3' end. So this is the first critical contact. Second contact is made with DSRBD, which is from non-canonical side, so it's not via helices but via the beta sheets, and the final interaction between loop and the L2I domain RNA. If you look at the respective positions of the substrate and of the catalytic sites, you can see that it's definitely not a cleavage competent state. So this is why we call it inactive state or closed state. We did the same analysis with all-site dicer and incomplete with the same microRNA precursor. And you can see from the first side, something is completely different. And that is the missing density for the helicase module and for the 2HP. So if you look at the structure, what we can see is that the RNA is in a different position, but this time it's bound in the bus domain where it's 3 and 5' ends, and it's bound into the catalytic pocket. And what we also can see is that there was rearrangement of the DSRBD, and this time it's contacting the substrate from the correct side, or from the canonical side. If you look at the relative positions of the cleavage site and the catalytic site, we can see that in case of RNase3A, it's reasonable distance, and in case of the RNase3B, again, it's kind of corresponding to what we would like to see. So that's why we call this cleavage competent or active state. So the question is, what are the implications for RNAi, so for the RNA interface? If we try to model long double-stranded RNA substrate into the closed state, we can see that there is a spherical clash with the helicase, and therefore it probably doesn't make much sense. If we do the same with the all-site isoform, what we can see is that we can, without any clashes, just trade it through. So this tells us that in case of all-site dicer, it's possible to just directly load the substrate into the catalytic site, while in this closed state, the dicer performs something like substrate selection. And if you look at the distance between bus domain and helicase, it would correspond to more or less 55 to 65 nucleotide long substrate. And if we look at the histogram of lengths or average lengths of microRNA products in mice, microRNA precursors in mice, we can see that most of them actually are in this interval. So now we see that definitely the Hellman domain plays a role in locking the dicer in this closed state to help it with substrate selection. So in order to find the mechanism by which the L1 domain actually does this, we looked at this interface between L1 and RNA's 3B domain and identified a handful of interactions. If you look at the sites, we can see that most of these interactions are actually rely on high-degree interactions and very often also on aromatics. So we performed primary sequence alignments, and to our surprise, we realized that most of the crucial residues in both of these domains are actually very nicely conserved in vertebrates, while being rather random in invertebrates. So this drives the question: what happens if we mutate them? So our collaborators again took initiative and created and took these 5 residues from the L1 domain and replaced them with either corresponding residues from the Drosophila, or for full lysines, or exchanged even the whole L1 for L1 from the Drosophila. And then they monitored RNA activity using some luciferase reporter assay. In this assay, the lower signal indicates higher RNA activity. So as we can see, all of these mutants have kind of similar signal to the delta L1, which is corresponding to the oocyte isolate. Even more surprisingly, this H83C mutant, which is just single point mutant, showed quite increased activity, which means that if even single point mutation can cause this, that this interaction is probably prone to be disrupted. So we wanted to see this, see the open complex for the full-length dicer, because for the full-length dicer we always observe just the closed state. So in order to do this, we employed the RBP2. It's a short protein consisting of 3 VSRBPs, and we just added it to the mix during the complex formation. This time we got, again, the majority of the particles gave us something that looked like the precleavage state or the closed state, while we got something else that resembles the, like 10 to 15%, something that resembles the open state. So we saw the structure of the majority of the particles. So we got 3.8 angstrom resolution map, where we could nicely fit the dicer, RNA, and we could also locate all 3 VSRBDs of the TRBP. When we tried to reconstruct the rest of the particles, what we observed was again missing density for L1 and that domain. And when we compared the structure that we built into this low resolution model with the previously shown oocyte dicer incompressive primitive DNA, we can see that they are really identical, which means that the RBP helped to push the dicer from closed to open state. So to put all this together, we proposed that mammalian dicer, the L1 domain locks the helicase in the closed state, and if too long double-stranded RNA binds into the bound domain, due to this spherical clashes, it cannot form stable complex. So it's released unprocessed. However, once the dicer is provided with some substrate of foreground, it can form the stable closed complex. So it passes the substrate selection step. Then comes the activation part or step, which means disruption of this interaction of this interface, which leads into the flexibility, or high flexibility of the helicase domain, which in combination with DSRBD rearrangement allows the RNA to actually lock into that open state. So now in the cleavage competent state, the available substrate is cleaved, and then the RNA product is released, and the dicer adapts back the original closed state. Yeah. So with this, I hope that I convinced you today that in mammals, the L1 domain adapted some helicase unrelated function in substrate selection, and that, this is what actually directs the selectivity or rather restricts the dicer to microRNA pathway. Furthermore, we also saw that microRNA has to fit into the very conscious protein, so most probably the evolution is on the side of the microRNA precursors and not on the side of the protein. The TRBP2 serves as allosteric activator, so the transition between closed and open state. And that lies the structural basis for reactivation of RNA-iron. So with this, I would like to thank to all our collaborators, to Mr. Sobura, to our Cryo-EM facility, and also to my group, so my boss, and especially Martina Donova, who did a lot of work in model building. Also, I would like to thank to all the funding from University and CETEC, CIISD, and Instruct for providing me this opportunity to speak. And with this, I would like to thank you for your attention.